Hello again. Let's complete with Module 6, Lesson 2, Reactions Energy. Let's start with the Part 2, Enzymes. What do you mean by enzymes? Enzymes in biology are important molecules that facilitate the reactions and reduce the time that is needed for the reaction. Let's start with the enzymes. All living things are driven by chemical reactions. That's known. Additional substances are needed to reduce the activation energy. In the previous lecture, we said that there is something called activation energy, and we said there is specific reactions called exothermic and specific reactions called endothermic reactions. These enzymes are needed to reduce the activation energy to start the reaction and to facilitate the reaction. Let's start with the catalysts. What do you mean by catalyst? Is a substance that lowers the activation energy needed to start a chemical reaction. Catalysts do not change the amount of a product produced, for sure, and they can't be used during the reaction. So the catalysts are a type of enzymes that facilitate and lower the activation energy, okay? At the same time, catalysts do not change the amount of a product that will be produced, and they will not be used during the reaction, okay? Special proteins called enzymes are the biological catalysts that speed up the rate of chemical reactions in the biological processes. So again, there is something called enzyme, which is special proteins. So proteins, as you know, there is many types of protein. One of these proteins is called enzymes. Enzymes facilitate and catalyze the reaction, which speed up the rate of the reaction and lower the activation energy. Most enzymes are specific to one reaction. So these enzymes are specific. They will not be used again in another different reaction. They will be used in specific type of reaction. Look at this energy diagram. In this energy diagram, we will just study first if this reaction is endothermic or exothermic. What do you think? This is endothermic or exothermic? Look at the reactant and the product energy. As you can see, the reactant energy is higher than the product energy. So this is an exothermic. So this is an exothermic reaction, okay? So we know that this is an exothermic reaction. It releases energy. Look at the colors of the diagram. We have two colors, the orange one and the green one. The orange one is showing the energy during the reaction without an enzyme, while the green one is showing the energy during the reaction while the enzyme is found in the reaction. As you can see, there is a huge difference. Look at the amount of energy here. There is a lower energy that is needed low energy that is needed for the activation. So as you can see here in this reaction, the amount of energy that is needed to start the reaction in the presence of enzyme is lower than the amount of energy that is needed in the absence of the enzymes, okay? So this is one of the importance one of the important functions of the enzyme, which is lowering the activation energy of any reaction. The reactants that bind to an enzyme, we call them substrate. So look at this picture. In this picture, you can find or you can see the enzyme, okay? And you can find something called substrate. What do you mean by substrate? Substrate is the reactant that will bind to the enzyme. For each enzyme, there is a specific substrate, okay? The specific location where a substrate will bind to the enzyme, we call it active site. So this is the active site. Again, this is the active site. So the active site for the enzyme where the substrate binds, okay? For each enzyme, there is a specific active site shape. It will not fit all of the substrate for sure, or all of the reactants. So for each enzyme, there is a specific substrate and specific reactant. Once the substrate bind to the enzyme, it will facilitate the reaction, and then the product will be released and the enzyme will be relaxed again. It will not be used during the reaction and it will be free. Okay? There is many factors that will affect the reactions, and it will affect the enzymes. Like what? Like pH, temperature, and other substances that affect the activity of the enzymes. Again, let's check these factors again. Each enzyme has a range of conditions in which it is most active. Factors that affect enzymes' activity including pH and temperature. Substances called inhibitors that can bind to an enzyme's active site, preventing the substrate from binding to the enzyme and slowing the rate of the reaction. There is something called inhibitors everywhere, صح? The inhibitor known to bind to the enzyme active site, and it will inhibit the substrate and the reactant from binding to the enzymes. Then what will happen? If the inhibitor binds to the enzyme, it will block the enzyme and it will not allow the substrate or the reactant to bind to the enzyme, and that will block the enzyme and at the same time block the reaction to happen. Okay? And once there is no enzyme, it will slow the rate of reaction. Who can give me an example for the enzymes that we can find it in our body? There is many enzymes that we can find it in our body, like the enzymes that will be used during the process of DNA replication, RNA transcription and translation. There is many reactions that we use it. Our body use it, especially in the enzyme that will be used to break down food. You know, when you eat food, the food will be broken down. There is many and multiple enzymes that will bind to that food to degrade and break down the food. As an examples of these enzymes, we have enzymes that found in the stomach, okay, like pepsin or pepsinogen, and we have enzymes that's found during the replication process of the DNA, like DNA polymerase, ligase, and there is multiple enzymes that found in the RNA transcription translation, like RNA polymerase, catalase, and so on. Okay, so there is multiple types of enzymes that we found it, or we can find it in our body. Okay, the enzymes, there is a huge number of enzymes, and these enzymes are needed for each reaction that happens in our body. Let's now do the quiz. Which of the following is a substrate that lowers the activation energy? Which of the following is a substance that lowers the activation energy? We are saying the substance that lower the activation energy. What do you think? Is it ion, reactant, catalyst, or substrate? The answer is catalyst. That's amazing. Which statement is true of chemical equations? Chemical equations. Reactants are on the right. Products have fewer atoms than reactant. Products are on the right. Reactants have fewer atoms than products. What do you think? For sure, let's look at the C and D. Both of them are incorrect because there is no change on the number of atoms on the reactant and the product sides. So what do you think? Reactants are on the right or products are on the right? What we can find on the right side? That's amazing. Products, for sure. Thank you. Which describes an exothermic reaction? Exothermic reaction. A, release energy. B, require energy. C, create energy. And D, destroy energy. What do you think? Can we create energy or can we destroy energy? For sure, no. So, either release or require. From the name exothermic, that release energy. That's amazing. In which of the following are bonds broken and new bonds forms? Is it in a chemical reactions, isotopes, elements with the polar molecules? It's chemical reaction. And the last question here, which of the following decreases enzymes activity by binding on the active site? Binding on the active site, is it inhibitors, catalysts, substrate, or pH? What do you think? For me, it's for sure inhibitors. And that's it. Thank you. Have a nice day.